Welcome back, students. Today we are going to discuss part 2 of Unit 9 of Application of Biology chapter. Now, before start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In today's video lecture, we are mainly discussing about these 4 points, means uses of microbes in sewage treatment, uses of microbes in the production of biogas, uses of microbes as a biocontrol agent, and uses of microbes as biofertilizer. So, first point, microbes in sewage treatment. As you know that the major component of sewage waste is human excreta, right? And this municipal wastewater is, that's why we can use the term sewage for the same. For what? For municipal wastewater. As this particular wastewater or sewage contain large amount of organic matter and microbes. This wastewater contains large amount of organic waste and water. So before disposal this sewage, we have to treat this sewage in STP or sewage treatment plant. Why? To make it less polluting. Okay, to make it less polluting. Okay, so as you can see, this is an aerial view of a sewage treatment plant. And this treatment of sewage water or wastewater is done by heterotrophic microbes naturally, which are already present in the sewage. And this treatment is carried out in 2 different stages. First one is the primary treatment and second one is the secondary treatment, or we can say biological treatment. Okay, so primary treatment. So in primary treatment, as you can see, there are 2 chambers which are mainly involved in primary treatment. So what is the first step? First step is the physical removal of small and large particles which are present in the sewage. Okay, so physical removal of particle from the sewage. How? Through filtration and through sedimentation. Okay, so initially floating debris is removed by sequential filtration method. Here only just removal of large big stone soil and big stone particles. Okay, through filtration and through grit, removal of soil and small stone or small pebbles are removed by sedimentation. Okay, so here just removal of through this screen, removal of big particle, big physical removal of big stone particles, floating debris and other things, right? And then removal of soil and small pebbles occur in this grit. And then what happened when all the solid that settle down in grit chambers, the upper part contains only supernatant part, this. And then this supernatant part, or we can use the term effluent for the same, it passes from the grit chamber to sedimentation tank side. Right? So all solid that settle from the primary sludge and the supernatant from the effluent. After settling down the all solid particle, the remaining part is, now we can use the term that remaining part, the term effluent for the same, right? So this effluent from the primary settling tank is taken for the secondary treatment. Okay, so the effluent from the primary settling tank, this is the sedimentation tank. All the solid particles, soil and other things are settled down over here. First at this point, then secondary at this point, and after settling down, this is transfer from grit chamber to sedimentation tank, and the upper part is act as a effluent. Now this effluent just move from the primary treatment towards the secondary treatment chambers. Okay, now as you can see, this is the big picture of sewage treatment plant. Just if this is the wastewater or sewage water, just cross different stages or different chambers of primary treatment for the removal of large particles, soil, small particles through debris screening and grit removal, as just now we have discussed, and then sludge, small particles is settled down in this primary clarification, and from here effluent is just passed from the primary treatment plant to secondary treatment plant, right? And in this secondary treatment plant, as you can see, this is known as aeration tank. First chamber is known as aeration tank. Okay, so we can now understand the term. In this particular tank, aerated or input of air, continuous input of air is going on in this particular chamber. Okay, so and after aeration, so the bacteria get activated and it continuously degrade or utilize the organic component present in this particular effluent, right? And after aeration, after the main work has been done, this effluent then pass from this aeration tank to secondary clarification tank, where some amount of sludge is settled down, a small amount of sludge is again processed towards the this aeration tank as a inoculum, and this time we can use the term activated sludge for the same, that act as a inoculum for the bacteria and fungus, right? All bacteria, fungus, both are present in this particular aeration tank, and they utilize the organic component for digestion of organic component, and remaining sludge is settled down into this sludge digester. After secondary clarification, this effluent again then pass through filters and disinfected through the UV lights, and then this treated effluent then pass, or we can say discharge, to the surface water. So these are the steps of sewage treatment plant. These are the steps mainly involved in sewage treatment plant. First one is the primary section, where removal of debris, big particle, small particles, soil occur, and digestion or biological. That's why secondary treatment is also known as biological treatment, because mainly bacteria and fungus strain involved over here in this aeration tank to digest the effluent. Okay, and then final screening occur at this third step, is which is known as tertiary treatment. Okay, so after treating the UV light or chlorine, it will then discharge to the surface water. Okay, right now we have discussed about the primary treatment. Now let's discuss the secondary treatment or biological treatment. Okay, this is the zoom in picture of the same picture which just now we have discussed. Okay, so now onwards, at this stage onwards, secondary treatment. All the tanks which are mainly involved in the secondary treatment are mainly I have shown that picture only. Okay, so what is written over here? The primary effluent is passed, this primary effluent. Okay, this is the part of primary treatment plant. Okay, so this primary effluent is passed into the large aeration tank. This is the large aeration tank, right? Where it is constantly agitated mechanically, and air is pumped into it. Why? Because this air allows vigorous growth of the microbes, which is already present in the sewage. Okay, so this air allows vigorous growth of useful aerobic microbes into the flocks, and this microbes means bacteria as well as fungus. So this microbe contain both masses of bacteria as well as fungal filaments. Okay, while growing, these microbes consume major part of the organic matter which is already present in the effluent. For their growth, bacteria need some carbon source, right? And that organic component, that carbon source already present in the effluent. So for their growth, we have provided air, है ना? And other organic component that is required for their own growth of bacteria and fungus. This is already present in the effluent. So microbes consume major part of the organic matter present in the effluent. And then what will happen? Because of the consumption of organic matter, it will significantly reduce BOD of the effluent, means biological oxygen demand, because most of the oxygen present in that particular effluent is utilized by the bacteria and fungus for their growth. Okay, so when they utilize this oxygen, so significantly this process reduce the BOD of the effluent. So what is BOD? What is the definition of BOD? BOD refer to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by water, by bacteria. Amount of oxygen that would be consumed, fully consumed, maximum amount of oxygen that would be consumed by the bacteria. Okay, by the bacteria. Okay, so sewage water is treated. At what time sewage water treated? Till the BOD is reduced. That time we can say, okay, if BOD is reduced, means now or maximum amount of organic component which is present in the effluent is digested by the microbes. Okay, so it is a test measure. BOD test, it is a test measure. This BOD test actually tells us the rate of uptake of oxygen by the microorganism in a sample of water. Thus, indirectly, this BOD tells us the organic matter, how much organic matter now present into the water. If BOD is high, means very much amount of organic component is present in any wastewater, and very less bacteria and or microbe is present. But when the number of bacteria and number of fungus continuously increase and doubling in their size, they continuously utilize the dissolved oxygen. So after some time, BOD is reduced. Okay, and that time we can say now most of the sewage water is treated by the bacteria. Okay, clear? So this is a test which indirectly tell us the measure of the organic component present into the water. Okay, so the greater the BOD value, means BOD is more, means we can say water is more polluted. BOD is less, then we can say BOD is less or BOD is reduced, then we can say now it is treated one. Okay, so once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into the settling tank where bacterial flocks are allowed to sediment. Okay, bacterial flocks are allowed to sediment, and this sediment is called activated sludge after treating in agitation tank. Okay, so as you can see, this is again the same picture. This is the primary treatment where so many large pebbles, small pebbles, soil particles are removed. Here main activity occur in this aeration tank. Continuously growth of bacteria and fungal is going on in this particular aeration tank, and simultaneously after some time what will happen? BOD get reduced. Okay, so this is the settling tank in which a small amount of sludge is pumped back. A small amount of activated sludge is pumped back to the aeration tank like this activation sludge, and this act as a inoculum. Inoculum of what? Inoculum contains bacteria as well as fungal strains. Okay, the remaining part of this sludge is settled down in this sludge digester. So remaining major part of the sludge is pumped into the large tank called anaerobic sludge digester. Okay, other kind of bacteria which grow anaerobically means that those bacteria which do not require oxygen for their growth, they actually digest the bacteria and fungus which are present in this sludge. Okay, so they digest bacteria and fungus strain which are present in this particular sludge. And during this digestion process, bacteria produce a mixture of gases during digestion, such as methane, such as hydrogen sulfide, carbon dioxide, and collectively we can use biogas term for the same. Okay, because these gas can use as a source of energy, as these are inflammable in nature. They can very quickly catch fire. Okay, so the effluent from the secondary treatment plant is generally released into the natural water bodies after tertiary treatment, means ultraviolet rays and other chlorine treatment. Okay, so the effluent of the secondary treatment plant is generally released into the natural water bodies like different kind of river or stream. And the Ministry of Environment and Forest recently, this Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save these major holy river of our country from pollution. Okay, under these plans, it is proposed to build large amount of sewage treatment plant so that only treated sewage may be discharged into the river. Okay, this is the main motto of these plants. Now, second main objective of today's video lecture is use of microbes in the production of biogas. So what is biogas? It is a mixture of gases. What is the main gas? CH4. Okay, CH4, H2S, carbon dioxide, these are the main gases. So biogas is a mixture of gases which are produced by microbial activity, which may be used as a fuel. Certain bacteria which grow anaerobically. Just now we have discussed this term anaerobically. Okay. So certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with carbon dioxide and hydrogen. Okay. And these bacteria are collectively called methanogens. Methanobacteria. These can grow anaerobically. These can, that's why after digestion of cellulosic material, they can also release this kind of gases which utilized as a biogas. Okay, so these bacteria are commonly found in anaerobic sludge also during sewage treatment, which just now we have discussed. As you can see, this is a picture of biogas unit. Okay, this is a from where mixture of gobar and other cellulosic material passed into this particular biogas unit, and from here after digestion, this is the gas outlet. Biogas is from this point of view, this is a mixing tank, this is the overflow tank. If content is more, then it will come out from this direction, and this is the digestion tank where digestion of that cellulosic material going on. Okay, so these bacteria are mainly present in these methanogen bacteria, anaerobic bacteria known as methanogen, right? So these bacteria are present in the rumen, means What is rumen? It is a part of stomach present in the cattle, means cow, buffalo, and these type of animals. Okay, a lot of cellulosic material present in the food of cattle, right? And is also present in the rumen. So in rumen, these bacteria actually help in the breakdown of cellulose and play an important role in the nutrition of cattle. And that's why their excreta, these cattle excreta, which is known as dung or gobar, right? That gobar also contain these methanogens. Okay, so this dung can be used for the generation of biogas, which is commonly known as gobar gas. So cattle dung is available in large amount in rural area. Why rural area? Because mostly cattle are present there, because cattle are used for the variety of agriculture purpose. Okay, so biogas plant are more often built in rural area, and the biogas thus produced is used for cooking as well as lightening of that village or rural environment, rural area. Okay, and this technology, biogas production technology was developed by IARI, Indian Agriculture Research Institute, and Khadi and Village Industrial Commission. These two main institutes involved in this particular to develop this particular technology, biogas production. Okay, now third point that we are going to discuss in today's lecture is use of microbes as a biocontrol agent. Okay, so first of all, what is the meaning of this biocontrol? Biocontrol refers to the use of biological method for controlling plant disease, just to control plant disease and pest, which mainly destroy any type of fasal, in which so many efforts and so many money already incorporated by the farmers, right? So biocontrol means use of biological method for controlling plant disease, and for controlling pest. Okay, in modern society, this problem has been tackled increasingly by the use of chemicals, by the use of insecticide, by the use of pesticides. But these chemicals are very toxic and extremely harmful, not only for human but other animals also. Okay, but also for our environment. Environment means for our soil, for our groundwater, for our fruit, vegetables, crop plant. Okay, because ultimately if we continuously use chemical, insecticide and pesticides on our various vegetables or cereals or crop plants, so continuous use will increase their concentration in the soil, in the water, and in the fruit, vegetable as well. Okay, so that's why these are extremely harmful for us and for our environment. Okay, if we continuously use weedicide for the removal of weed from our soil, that will also increase their concentration in the soil. Now next is biological control of pest and disease. So in agriculture, there is a method of controlling pest that relies on natural predation. Okay, rather than introduced chemicals. And this is known as biological control of pest and related disease. Okay, so what will happen in this method? The organic farmers actually works to create a system where insects that are sometimes called pest are not eradicated. Why? Because sometimes they act as a useful tool. How, we will discuss very soon. Okay, so these organic farmer kept all those pests at a manageable level by a complex system of checks and balance within the ecosystem. Okay, so the use of biocontrol measures will greatly reduce our dependency on toxic chemicals and pesticides. An important part of biological farming approach is to become familiar with the various life forms that inhabited in the field, various predator as well as prey, and also their life cycles, pattern of feeding, and the habitat that they prefer. Okay, so what is the main objective? Main objective to create a system where insects that sometimes called pests are not eradicated. Rather, these organic farmers use these kind of insects to eradicate other insects. How? Let's discuss. I have given you two examples. First one is ladybird beetle. Okay, this very familiar beetle with red and black marking. And the second example is dragonfly. Okay, these insects are useful to get rid of aphids and mosquitoes from the environment. Okay, similarly, to control butterfly caterpillars is the bacteria act as a biocontrol agent. Which bacteria? Bacillus thuringiensis. As this Bacillus thuringiensis or BT is highly selective. This Bacillus thuringiensis is highly selective. It only kills caterpillar and is not toxic to their natural enemy. Okay, this bacteria is harmless to human and animals and is considered environmentally safe. Okay, these bacteria are available in sachet as dry spores, and the farmer just mix these sachets with the water, content present in this sachet with the water, and spray that solution on the valuable plants such as brassica, such as some fruit trees, which these are where they are eaten by the insect larva. Okay, so in the gut of larva, the toxin is released and larva get killed. And this toxin is released by whom? That particular bacteria. Okay, so the bacterial disease will kill the caterpillar but leave the other insects unharmful because they are not the natural enemy of other insects. Okay, because of the development method of genetic engineering in last decades, the scientist has introduced this Bacillus thuringiensis toxic gene into the plant as well. So they develop genetically modified crop like BT cotton, BT corn. Okay, such plants are resistant to attack by insect pest. BT cotton is one of the such example, as you can see in this picture. This is the genetically modified BT cotton and BT corn. Okay, so these modified plants are resistant to attack by insect pest. So a biological control being also developed for the use of in the treatment of plant disease is the fungus. We have discussed various type of pest. We have discussed bacteria as a biocontrol agent. Now fungus as a biocontrol agent. So here trichoderma is the example. Okay, this trichoderma species are free living fungi that are very common in the root ecosystem. They are effective biocontrol agent for several plant pathogens. Okay, similarly, virus, baculovirus. These are the pathogens that attack insects and other arthropods. Okay, so majority of baculovirus used as a biological control agent in the agents are in belong to the genus Nucleopolyhedra-virus. Nucleopolyhedrovirus. Okay, so fungus as a biological agent, virus as a biological agent, bacteria, this Bacillus thuringiensis as a biological agent. Okay, some insects like lady bird beetle, dragonfly as a biological agent. So this is the main principle of using biological material or biological life forms to control the pest. Okay, now next point, uses of microbe as a biofertilizer. Okay, so biofertilizer, what is biofertilizer? Biofertilizers are organisms that enrich the nutrient quality of the soil. Okay, the main source of biofertilizers are bacteria, fungus, and cyanobacteria. First, bacteria, fungi, and cyanobacteria. These are the main sources which act as a biofertilizer. As you can see in this picture also, there are root nodules present in the root. Okay, so these root nodules is present in the leguminous plants, and this nodule present in the leguminous plant, this contained rhizobium and this rhizobium bacteria shows symbiotic relationship with the leguminous plant. Okay, as these bacteria actually fix atmospheric nitrogen into organic form and plant provide them nutrition. So they have a symbiotic relationship, right? Other bacteria also fix atmospheric nitrogen, while free living bacteria which are present act as a free living in the soil, like azospirillum, like azotobacter, and thus enriching nitrogen content into the soil. Okay, but rhizobium act as a biofertilizer, mainly for these leguminous plant. Okay, similarly fungus. Some fungi also show symbiotic relationship with some plant roots, like mainly in cycas, and that relationship is known as mycorrhiza. Okay, and most of the members of this type of fungi is associated with the genus Glomus. This fungus symbiont, in this association, they actually help plant to absorb, or they provide phosphorus to the plant from the soil. Okay, and plant provide them nourishment. Okay, so again a symbiotic relationship between fungus and plants. So plant having such association, this kind of symbiotic relationship, other than phosphorus, they also get other benefits like resistance to root-borne pathogens, tolerance to salinity and drought, and overall increase in plant growth and development. Okay, so this is the second example. Third one is cyanobacteria, which act as a biofertilizer. Cyanobacteria also fix atmospheric nitrogen. Okay, and what are those examples? Examples include in the cyanobacteria Anabaena, non-stalk, oscillatoria. These are the examples. Okay, so in paddy field, means rice field, cyanobacteria serve as an important biofertilizer. Okay, this blue-green algae or cyanobacteria add organic matter to the soil and increase its fertility. So mainly in rice field, blue-green algae act as a biofertilizer. Currently in our country, a number of biofertilizers are available commercially in the market, and farmers use these regularly in their field to replenish soil nutrient and to reduce dependency on chemical fertilizer. Okay. Now, biopesticides. Okay, so biopesticides is a biological substance or organism. It may be a substance or organism that damage, kill, or repel organisms seen as a pest. Okay, biological pest management intervention involve predatory, parasitic, or chemical relationship. They are obtained from the organism. They may be some kind of extract. Okay, or so they may be obtained from organism including plant, including bacteria, or some fungus or nematodes. Okay, so what are the examples? These bio-driven chemicals include pyrethrum, rotenone, neem oil. These are the examples. Okay, these are the extracts obtained from various sources, plant, bacteria, fungus, etc. Okay, so these are the examples. First is pyrethrum, rotenone, neem oil, and various essential oil. These are naturally occurring substances that control pest and microbial diseases. Okay, so these are all of today's topic of discussion. I hope most of the things has been cleared now. Still you have any doubt, any confusion related to today's video lecture, please let me know and write down in the comment box. We will discuss it later on. Till then, take care. Bye bye. Keep learning. Namaskar.